Today we will discuss about Calnexin and calreticulum cycle mediates the ER quality control. In the previous video we have discussed that Calnexin and calreticulum are essential for the deacylation of these GPI-APs, or we can say that it is essential for properly folding of the GPI-APs that are translated in the endoplasmic reticulum. In this video we will use this CD59 to check whether this Calnexin and calreticulum uh, helps in maintaining the ER quality control or not. So they have used the mutated CD15. So let's go into the experimental results. So what they have done is they have used two different reporters. First one is EGFP reporter, that is EGFP flag reporter that is attached to this CD59, mutated CD59, where C94 is mutated with S, and simultaneously they have used this KDL reporter, which represents the proteins that are expressed in the endoplasmic reticulum, and this is the merge image. And for the wild type, they can see that there is an overlap of this EGFP signals and this KDL sequence, indicating that most of this mutated CD59 are within the endoplasmic reticulum and are not expressed on the plasma membrane. And same things are been quantified in the panel D using Pearson correlation coefficient, where one indicates that there is a complete or 100% overlap of this EGFP and KDL sequence. For the wild type, it gives a signals of around 0.715. And when they check this same results for the calreticulum knockout and calnexin knockout, they can see that there is also they are able to find that there is an overlap between EGFP signals and KDL sequence. There is no statistical difference between non-overlap of this EGFP and KDL sequence between calnexin or calreticulum knockout to the wild type cells. So this from this we can make a point that quality control within the calnexin and calreticulum knockout is in line with the wild type. However, for the dual knockout, they can see that there is a complete disoverlap, or the signals for the EGFP are not overlapped with the KDL, indicating that there is a quality control loss, and most of these mutated CD59 are expressed on the plasma membrane. And these things are also been validated using this Pearson correlation coefficient, where the non-overlap of this EGFP and KDL sequence is statistically significant compared to the wild type, and it is represented by this blue column. And after validating that it's a dual knockout, there is a quality control loss occurs, then you check whether if these carbohydrate structures are not monoglucosylated, then also it is able to interact with the calnexin and calreticulum or not. So for this they use this knockout, this GANAP-KO and MOX-KO, and they help in trimming of the glucose whenever they are translated in the endoplasmic reticulum. So if these cells with these knockout cells won't be able to cleave the glucose, and they should not be able to interact with the calnexin and calreticulum as per the literature. And in this case, when they check the signals of this EGFP and KDL, they can see that there is some kind of non-overlap occurs that can be seen here, indicating that most of the proteins are retained within the endoplasmic reticulum. However, some of them are able to interact with the calnexin and calreticulum and can express on the plasma membrane. And same is the case they have observed for the MOX knockout also. And these things are also reflected in the quantitative analysis of this fluorescence images, where they can see that the difference between there is a statistical difference in the GANAP knockout overlap and MOX knockout overlap compared to the wild type. And after validating that the monoglucosylated structures are necessary for calnexin and calreticulums, they also need to check if there is a non-glycosylated proteins are also required. Non-glycosylated proteins also require this calnexin and calreticulum cycles for the ER retention, or we can say that for proper folding. So they mutated the CD59 glycosylation site, that is N43 is mutated with Q and C94 is mutated with S. And when they check the signals for this EGFP and KDL, they can see that there is a non-overlap, indicating that non-glucosylated structures do not interact with the calnexin and calreticulum, and they will move to their final destination locations. And same is the case they have observed for this dual knockout also, that there is a non-overlap of this EGFP signal and KDL sequence, and these things are reflected in this Pearson correlation coefficient analysis of the fluorescence images also, where the results are statistically significant, are decreased, and these decrease are statistically significant not only with respect to wild types, but the wild type with CD59 mutant, but also the there is a statistical difference between the signals non-glycosylated structures that are retained in the endoplasmic reticulum in the wild type cells and also in the dual knockout cells. These differences are also statistically significant, indicating that there is a higher non-glucosylated structures that expressed on the cell surface compared to the case of these wild type cells. And so let me repeat my last statement. So there is a higher non-glucosylated structure expressed on the cell surface in dual knockout compared to the case of wild type cells. So there is a more quality control loss when in the dual knockout. This is what it means to say. So I hope you understood this, how this calnexin and calreticulum cycles works with the example of this CD59 using different mutations. First one is by mutating it so that it can acquire the misfolded structure. Second one is by using the cells that does not have monoglucosylated carbohydrate structures formation ability. And third one is by mutating the proteins such as CD59 so that it is not be able to glycosylate it in the endoplasmic reticulum. If you need to understand more about their experimental results in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.